Thank you. To come back to your question, Rafa, actually there was a newsletter published a couple of weeks ago, I forgot to include in my slides, but showing that now in Europe the number of industry-manufactured CAR T cells is exceeding the number of academia-manufactured CAR T cells that are being used. So I think the industry part is taking over, which does not mean that we should not initiate more academia-driven trials, of course. Anyway, so I will conclude this session and I think make a nice transition after Alvaro's presentation and Michael's presentations by going over the hurdles to initiate CAR T activity in relation to the industry. So these are my disclosures. I think that actually working with the industry creates a new form of conflict of interest because you become a supplier of human-derived elements as a starting material to manufacture a medicinal product that, as was already mentioned on several occasions, is quite costly at the end when it comes back to your hospital to be administered to your patients. Anyway, I won't go into the details, but let's consider practical issues. What are CAR T cells from a biological perspective? We heard that these are living cells that are genetically engineered. This is a highly personalized therapy, at least in the autologous form. From an engineering perspective, that comes as cryopreserved cell suspension, most often in plastic bags, although the container can vary from one manufacturer to the other, and the product manufactured by Juno and Celgene, for example, is significantly different from the products manufactured by Novartis and Gilead. And from a regulatory perspective, this is a gene therapy medicinal product. This is a subcategory of advanced medicinal products as defined in the 2007 European regulation. And these are also genetically modified organisms similar to maize, for example, manufactured by, let's say, Monsanto. There are some implications in those keywords. They are manufactured from human living cells, and as already mentioned, most of the currently available CAR T cells are of autologous nature. Cell procurement happens on an individual basis from sick patients, and the service is provided by hospital or in some cases by third parties such as other hospitals than the one treating the patient or possibly blood banks. These are medicinal products, so at least the approved products are manufactured in compliance with good manufacturing practices, and there is an adapted version for advanced therapy medicinal products, but in this presentation I won't go into the details, and the distribution is under the responsibility of hospital pharmacies. These are cryopreserved viable cells, so this comes as a fragile product. And the picture here is not a CAR T cells, but that's an old picture that we took from an autologous blood transplant. But this is what you don't want to happen. This is a broken bag getting out of the liquid nitrogen tank, and this could actually happen as well with CAR T cells. And obviously the implication for a product that is worth 350,000 euros are quite significant, and we need to define who is responsible in case this happens. And actually I am aware of at least one situation in which a CAR T cell bag was broken right at the time of infusion into the patient. So there are measures to be implemented to face that kind of situation, even if this happens quite rarely. But we need some technical expertise to safely handle and store these products. And this expertise at the moment is mostly present in cell processing facilities, not that much in hospital pharmacies, and we will need some time to properly train the hospital pharmacies and equip them with cryobiology equipment. And last, as mentioned before, these are genetically modified organisms. So all measures are taken to make sure that these products are free of replication-competent viruses, so the risk for the environment is very low, and regulatory agencies classify CAR T cells at the lowest level of risk for the environment, but still your hospital will need to take some measure and specific rules, in particular to discard waste generated throughout the distribution and infusion process for the CAR T cells. So at the moment we only have 2 products that have been approved, first in the US in 2017 and a year later in Europe by EMA at the end of August 2018. So we are basically 6 months after the first approvals in Europe. So Tisagen Leclecels, it's easier to say Kymriah, is manufactured by Novartis, and Axicaptagen Siloleucel or Axicel or Yescarta is manufactured and marketed by Kite Pharma and Gilead. And all of the CAR T cells at the moment are investigational products, as nicely described by Alvaro Urbano in the previous presentations, with a fair share of the trials being initiated by academic facilities such as London or Barcelona, as described previously. My concern about those trials is, and that was the last question I guess, where is that leading? Because some of these trials are really testing new products with improved CAR constructs, for example, but some of those trials are mostly replicating results of trials that have been conducted by the industry, and we all wonder how sustainable that will be in the future, because there is very little chance due to IP reasons that these products can be placed on the market, and unless competent authorities and regulatory authorities significantly change the regulatory framework, I personally doubt that in the future it will be easy for academic facilities to manufacture these products, but I'm having a rather provocative assertion there so that we can discuss that further after that. Anyway, let's focus on industry manufactured products for this particular talk. Both the authorized products, as we heard, target CD19, are autologous in nature, and are manufactured through viral transduction of immunoselected T lymphocytes collected from the peripheral blood of sick patients. So the starting material is composed of autologous blood mononuclear cells that are collected by apheresis from candidate patients. And I always use this illustration that was published in a short editorial accompanying to a major paper in the New England Journal of Medicine at the end of 2017, because they identify the first step where the cells are collected from the patient, and the second step when the drug product is returned to the hospital in a cryopreserved state and ready to be infused into the patients. So I won't cover the details of the manufacturing process because it's been nicely explained before by Michael, and there has been plenty of communications during this 3-day meeting on these topics. So procuring the starting material for the manufacturing process, again, the starting material is composed of peripheral blood mononuclear cells that are obtained through apheresis of patients treated for malignancies. So in terms of technical expertise for collection, the closest cell products that apheresis facilities are used to collect are allogeneic mononuclear cells that are collected from healthy donors for donor lymphocyte infusions. The main difference being that in those patients there is usually a significant lymphopenia as compared to healthy donors. There is no or little use of autologous mononuclear cells in the cell transplant practice. So in most apheresis facilities, the procedures needs to be set in place and validated specifically for the production of CAR T cells, of autologous CAR T cells. And then the collected autologous blood mononuclear cells will be shipped following the manufacturer's instructions. There are significant differences in the two processes meant to manufacture Yescarta and Kymria. The starting material is immediately shipped as fresh cells to Kite Gilead manufacturing plants for Yescarta, and for Kymria, the manufacturer asks the local facility first to freeze the cells before shipping the frozen product to the manufacturing plant. The cell shipment is usually under the responsibility of the cell processing facility, so the product will not be shipped directly from the apheresis facility but from the cell processing facility, because this is the tissue establishment, to use the wording that Europe regulatory bodies use to designate these infrastructures. Tissue establishments are the holders of the authorization to export human-derived elements, and in most cases there will be a cross-border shipment. For both companies, their main manufacturing plants at the moment are located in the US, on the West Coast for Gilead, as we heard yesterday, on the East Coast for Novartis, and we are all waiting for additional manufacturing capacities to come to Europe. But again, at the moment, the cells, most of the products will be shipped to the US, but through a European facility. So actually, you will ship your cells to a European site that next will ship it to the US. There is no clear recommendation on whether we should sample the collected cell product before shipping it to the manufacturing facility. That's what we do at my institution, because we consider that we are somewhat responsible for making sure that we ship product with the appropriate number of cells, and this, of course, is microbiologically negative. But you have to respect the manufacturer instructions to make sure that by sampling the product you don't contaminate it, for example. And there are uncertainties as to the technically easy step of manufacturing process for Kymria. Modalities for cryopreservation are significantly different from one side to the other. This is a recurrent problem with cell processing facilities. And as you know, that was already mentioned, but the devil is in details, and by slightly changing the composition of the cryopreservation solution, for example, you may significantly affect the biological properties of the shipped cell product. At this stage, the cell product is not a medicinal product, is not a drug product, and then you don't need to involve your hospital pharmacies. This is mostly business for collection facilities and processing facilities. And we definitely need clarification from the competent authorities on how they want to regulate this step, especially when the cell processing facility is requested to cryopreserve the cells before shipment. This is a minor step in the manufacturing processing, but it is still a step in the manufacturing processing. And I can tell you that in France, for example, it's very difficult at the moment to understand how the French competent authority wishes to regulate and potentially authorize centers to participate to this process. Manufacturing conditions, so GMP conditions, I won't go into the details of these boring requirements, regulatory requirements. But one important point to understand is that when you work under GMP conditions, you are supposed to audit all of your suppliers. And the point where I am coming is that, again, the hospital or the blood bank de facto becomes a supplier to the manufacturer. And this is one of the major reasons why, before starting to treat patients with CAR T cells, the company will come to your hospital and audit and evaluate how you are organized and how you will provide the service for cell collection and cell processing before the manufacturing process will start. So this comes to which criteria are used by pharma for center selection. And as stated on this slide, at the moment, these are mostly pharmas that decide which center they will qualify to produce and deliver their CAR T-cell products to the patients. So you go through an audit, which is quite similar, for those of you who know that, to audits conducted by competent authorities or inspections conducted by JC for those centers who have obtained JC accreditation. And actually, one of the first criteria that companies will ask for are whether or not you are JC accredited. And I believe that if you are, that greatly facilitates the qualification process for the companies and for the hospital. So the most relevant section in the JC standards is section C, that describes all the requirements for cell collection and apheresis, and to some extent section D, that describes the requirements for the cell processing facility. In addition, companies will ask for specific proofs of evidence for training of your personnel on their specific requirements, either for collection or for processing. And the companies will provide some of the necessary training, but some other aspects you will have basically to design by yourself and make sure again that you comply with all of their requirements. So this is the copy of the first page that you know of the FACT JC standards. Version 7 is the currently used version, and version 8.0 is in preparation, and I will come back later on the specific set of standards that is currently being designed to apply for immune effector cells administration. So the issues with center selections at the moment is that there is no harmonization in this process. So every company will come to your hospital and you will have to get through the qualification process for Gilead. You have to get through the qualification process for Novartis and for Celgene. And if you deliver other categories of gene therapies or somatic cell therapy products, maybe you work with Bluebird Bio and their globin disorder gene therapy, you will have to go through their own qualification process. So it's very much cumbersome, time-consuming, and for those hospitals who have been qualified since the two products were approved by EMA in mid-2018, people have spent hours with no planned compensation for this preparatory work. People have spent hours to get through the qualification process. And at the moment, we are also waiting for further indications from health authorities and competent authorities to oversee this process at a national and European level and make sure that patients get equitable access to these treatments regardless of the area where they live. And from the map that Alvaro showed in the previous presentations, we can see that there are some areas in Europe that are not covered at the moment, which means that patients have to travel long distances to get access to the treatments, and that need to be improved in the near future. Patient screening validation, not anything very specific. The rules derive from the tissues and cell directives that are more than 10 years old now, that are likely to be revised in the near future by the European Commission, but the revision has not been initiated yet. And practically speaking, the screening for communicable diseases is very much like for an autologous transplant, serological testing for HIV, hepatitis B, hepatitis C, syphilis and HTLV in most European countries, except otherwise defined by the study sponsor if we speak of an industry-sponsored trial. Specifications for the collected cell product, numbers of TNC to be collected, numbers of lymphocytes to be collected, that will be defined by the manufacturer, and you will have to demonstrate that you have implemented actions to meet these predefined specifications, actions in relation to the patient, or tailoring the procedural parameters for apheresis. Although it's fair to say that the manufacturing process for all companies that have approved products or products in development, the manufacturing process is quite robust and will be effective in the vast majority of cases, regardless of patient conditions, especially the lymphopenic status of the patient. I have identified 3 publications from American groups that have looked actually at the collected cell products and tried to identify criteria in relation to the patients or to the apheresis procedure to predict the quality of the collected cell product and how suitable this collected cell product would be for manufacturing. So you can see that there is a linear correlation between the pre-apheresis CD3 positive cells in the peripheral blood and the number of cells that you can collect in the bag. And for those of you who are familiar with apheresis, you can use a collection efficiency criteria which basically measure the proportion of circulating CD3 cells that you can collect through the apheresis process. And for the sake of time, I may skip the 2 other presentations. There is no specific program on the existing cell processors from the different manufacturers that you can use that are available in the market. So basically, you will use the same program and process that you use to collect allogeneic mononuclear cells for the preparation of DLI to collect the cells that are meant to manufacture CAR T cells. Scheduling, this is very important. As we heard before, there is a shortage of manufacturing capacities, mostly because the number of manufacturing plants is limited, and especially for Europe, there is few operating plants in Europe at the moment. We are expecting new manufacturing plants coming to several European countries in 2019 or 2020. So you cannot plan your apheresis by yourself. You have to really tightly coordinate with the manufacturer of the approved cell product, and in some cases the slot will be available only several days or several weeks after your intent to treat your patients. So that means also you need to tightly coordinate with the clinicians because bridging chemotherapy may be necessary for the patient to wait for the collection and for the manufacturing of the product. And we need more robust data, but as we heard before, the delay necessary to manufacture the product may be significantly longer than what has been reported in the original trials conducted in the US, both because we are in real life and also because shipping the cells across the Atlantic means a few more days before the final drug product comes back to your hospital. I won't really go into details because Michael already mentioned that even if you fully control all the steps of the manufacturing process, you will still have a quite variable product at the very end. This is an old publication, but showing in one of the first reported clinical trials how variable the product can be, even with a fully harmonized manufacturing process. Let's skip this one. So maybe I will go over the second part, which is how the products come back to the hospital in the afternoon session. Let me move to the last slide, first of all, to show you where we are today, where we could have been if I had convinced the organizers to have this meeting in Marseille. This is a really nice place, so I will lobby very hard to go there for the next edition. My group on the left-hand side of the slide, the clinical people and the patients. I think patients are very important to implement these new therapies. And for those of you who speak French or want to train with French, there is a special issue that was recently published by the French community on CAR T cells, where you can find a coverage of many aspects of the CAR T cells issues. Thank you for your attention.
Christian, thank you for this good overview and let us show the complexity of the process. So there is room for several short questions.
Hello.
Please.
Right at the back, Christian, Tony Pagliuca from London. Can I ask you, you mentioned supplier audits, and of course we're a supplier to the company, but the company are a supplier of product to us. So under standards, we should be auditing them. So should we be paying visits to their production facilities to ensure that the supply product is appropriate?
Should we get paid for that? Is that correct?
Well, not get paid, but just from the well, of course we'd like to be paid for it, but to have the time to do it. But legally, there may well be a requirement for us to audit the supplier of the product.
Yeah, but this is medicinal product. So as you know, the approval process basically is meant to guarantee the quality of the product. So when you buy a medicinal product, you don't audit the supplier. But the specific, so that's not my position. Let's make it clear. This is the official position of regulatory agencies that I'm just repeating there. I think the specificities with CAR T cells, these are highly personalized medicine, and as you perfectly know, we don't know exactly what we are infusing into the patients. We don't even know the exact dose of cells. And there are ongoing discussions at the national and European levels to request from the companies that they actually provide the physicians with the actual composition of the cell product that is infused. On top of that, an additional problem is that there is absolutely no QC that you can do on the product once it's thawed. So you are basically blind on what you infuse into the patient.
Herman?
Just an additional point. Actually, German inspectors went to the US and inspected the production side. So,
Interesting. Final comment?
Yes, I mean, you had been provocative by saying that probably the academic should not be implementing or developing more these sorts of CAR T cells. And I don't agree with that, because it's through the development of academy that innovation into the sorts of next generation, so better CAR T cell products will come. So I think it's a very important collaboration that we need to carry on that.
No, definitely, I think the academia should remain active. As I think that Michael showed the initial publications that actually came from Israeli and Japanese people, and most of the development, the commercial development, was done in the US. And the consequence is that now we, as European investigators and practitioners, we are paying very high costs for industry manufactured products. So I'm not at all saying stop developing academia. We should develop academia. Very happy that so many people were present in this meeting, and that actually we see that there is ongoing activity in Europe, and we expect it to be higher over the next years. Of course, academia should be there.
Very good. So thank you very much,
Thank
Christian. And I'd like to thank